most significant among these tools are the enzymes called endonucleases. Endonucleases are enzymes which break the DNA strand anywhere in the middle. In other words, they don't act at the terminal ends of the DNA chain, but can act in between, anywhere, breaking the DNA strand into fragments. Among these endonucleases, there is a particular group of endonucleases have been identified only in bacterial cells. They are known to be produced only in bacterial cells. These enzymes have been called as restriction endonucleases. Restriction endonucleases. This is because these enzymes cut the DNA molecule at specific sites, not randomly cut the DNA molecule at specific sites. What are these specific sites? These specific sites will be regions containing what are known as palindrome sequences. Regions containing palindrome sequences. Probably you would have heard of the word palindrome with reference to English. Palindrome is a word used to describe words which have the same spelling when you read them in both the directions. For example, look at this word look at this word rotator. R-O-T-A-T-O-R. From this side also it has the same spelling, R-O-T-A-T-O-R. So this is called as a palindrome. Similarly, to give you another example, the spelling of Malayalam. The spelling of Malayalam, M-A-L-A-Y-A-L-A-M. M-A-L-A-Y-A-L-A-M. So the word Malayalam is a palindrome. It need not be a word. It can be even a phrase. For example, look at this phrase, Madam, I am Adam. Madam, I am Adam. M-A-D-A-M, I am A-D-A-M. M-A-D-A-M, I am A-D-A-M. So such words and phrases in English are called palindromes. In the DNA molecule also we have sequences which read the same in the opposite directions between the 5' 3' end and the 3' 5' end. For example, in a portion of the DNA molecule, for example, between the 5' end and the 3' end, anywhere there may be a sequence like this, GAA TTC, GAA TCC somewhere. For this, in the complementary chain, in the 3' to 5' position, you will have CTT AAG. So you read this from 5' to 3' end, GAA TTC, GAA TTC. From the 3' end, CTT AAG. From the 3' end, CTT AAG. So this sequence, such a sequence is known as a palindrome sequence. A palindrome sequence. So restriction endonucleases identify such specific sequences in the DNA molecule and cut the DNA molecule in the region containing the palindrome sequences. That is the reason why they are known as restriction endonucleases. The first restriction endonuclease, they are abbreviated as REN. The first restriction endonuclease used was named as HIND2. There is a specific reason and way by which they are named. We will come back to that later. HIND2. And today the most widely used restriction endonuclease is EcoR1. Why such specific names are given, we shall see later. This restriction endonuclease has been obtained from the bacteria Escherichia coli. Escherichia coli, and is named as E cohort 1.